All right, welcome to my latest video series titled Is Evolution Biochemically Possible? Here I will be approaching evolution from the subject of biochemistry, which is the study of the molecules that make up living things, such as DNA or proteins. Let me make a few distinctions. By evolution, I mean macroevolution, which is the changing of organisms into a fundamentally different type of organism, such as going from dinosaur to bird. It does not refer to microevolution, which is the adaptation of organisms to their environment, which happens commonly. Also, I will be supporting the theory of intelligent design, which is that life cannot have formed randomly and needs a designer with a certain degree of intelligence. I am not arguing for creationism, which is a specific kind of intelligent design unique to Christianity that involves a six-day creation. Intelligent design is often mistaken for creationism, so I am making this distinction now. With that being said, this series is 6 parts. Part 1 will introduce the topic of evolution. Part 2 will explain why evolution should matter to Christians. Part 3 will address the biochemical evidence for evolution. Part 4 will introduce a biochemical argument against evolution called irreducible complexity. Part 5 will introduce the intelligent designer that comes from that, and Part 6 will summarize and propose some additional difficulties for evolution. Please note that some of my arguments were obtained from other sources, especially Michael Behe's book, Darwin's Black Box. I will cite those when appropriate. All right, welcome to Part 1, where we will introduce the topic of evolution. To understand evolution, it is necessary to understand some basic biochemical concepts. If these explanations are not sufficient, I would suggest looking through a biochemistry textbook or even searching these things on the internet, as this is fairly common knowledge in biochemistry. Starting with DNA. DNA stands for deoxyribonucleic acid, and it is the molecule that is the blueprint of every organism. So, just as a blueprint would tell an engineer how to build a car, DNA tells the body how to carry out its functions. A picture of DNA is included. It is shaped like a double helix, which is like two winding staircases wrapping around each other. DNA is incredibly small, so small that in humans, about 3 billion of its components can fit inside the nucleus of the average cell. DNA is stored inside the nucleus because it is safe there and it won't get damaged. That way, your blueprint stays the same. Also of relevance is that an organism's sequence of DNA is called its genotype. So humans have a genotype that is about 3 billion base pairs long. That's a bird's eye view. Let's zoom in on the molecule of DNA. DNA is made of components called nucleotides. So if DNA is the code, then the nucleotides are the individual characters in that code. It follows that a change in the code, or a change in even one nucleotide, can cause a change in the organism. These are called mutations. Let's zoom in even further. Each nucleotide contains a phosphate, a sugar, in this case deoxyribose, and a base. The DNA molecule looks like a set of stairs. The phosphate and the sugar form the railings of these stairs, and the base forms the steps. Each step you see is made of two bases bound to each other in the middle. These bases are adenine, guanine, cytosine, and thymine, abbreviated A, G, C, and T. A always binds to T, and G always binds to C. So each step is either made of A and T or G and C, and each half of the step is bound to its own sugar and phosphate in the railing. So a change in the DNA, called a mutation, would, for example, substitute an AT base pair for a GC base pair. Since DNA is the code, a change in the code can cause a change in the organism. DNA does not directly code for an organism. There are some intermediate steps. That is, DNA codes for proteins. Think of proteins as molecular machines that do just about everything that a cell needs to do. This includes interacting with the outside environment, breaking down food, reproducing, and many other functions. In this way, DNA is a blueprint for most organisms. That is, our DNA determines what proteins we build, and what proteins we build determines what functions we do. An example of a protein is hemoglobin, which is pictured here. Hemoglobin carries oxygen from our lungs to our body's tissues. There is a slight problem in building proteins from DNA, in that DNA is always kept in the nucleus, but the machinery to build the proteins is kept outside the nucleus. How does a cell bridge this gap? That is where messenger RNA comes in, abbreviated as mRNA. mRNA is a specific type of RNA, which stands for ribonucleic acid, which is similar to deoxyribonucleic acid. I gave a picture of RNA. It is pretty much the same as DNA, except that it is single-stranded, has an extra oxygen atom, and contains uracil instead of thymine. So, mRNA carries the DNA's instructions outside of the nucleus of the cell into the main body of the cell, where it can be used to build proteins. This is how mRNA gets its name. At least in eukaryotic organisms like ourselves, it is a messenger for DNA that carries DNA's instructions outside the nucleus so they can be used to build proteins. The process of going from DNA to mRNA is called transcription, and the process of using that mRNA to build proteins is called translation. There is more to come on this in a bit. So far, we have DNA, which is the blueprint, is transcribed into mRNA, which is translated into proteins. Proteins determine an organism's traits, which are its properties. For example, one trait of this finch might be that it has yellow and black feathers, or that it has a pointy beak. And the reason it has that trait is ultimately because its proteins built the beak that way. Traits can be internal or external. An external trait is the pointy beak, and an internal trait would be, for example, an increased percentage of red blood cells, known as hematocrit. So, DNA is transcribed to mRNA, which is used to build proteins, which determines an organism's traits. Traits, in turn, determine fitness, which is an organism's ability to reproduce. For example, maybe the fact that the finch has a pointy beak allows it to more effectively catch insects, thus increasing its fitness. That is, a finch that can catch more insects is more likely to survive than the finch that can't, so it is more likely to pass on its genes. Here is a figure representing transcription and translation. Remember how each A binds to a T and each G binds to a C? Well, in transcription, the DNA strand is split in the middle, exposing each base pair. Then the nucleotides of mRNA, which are mostly the same, are added to one of these half strands. Once the mRNA strand is made, it falls away and the DNA closes back up. This strand of mRNA then travels outside of the nucleus into the cytoplasm, where it binds to a machine called a ribosome. The ribosome basically uses the code in the mRNA to attach amino acids to one another, thus forming a protein. This process is called translation. Of course, which proteins are made influences traits, which influences fitness, and so on. So in summary, DNA determines mRNA, which determines proteins, which determines its traits, which determines fitness. So it follows that a change in an organism's DNA, called a mutation, can ultimately cause a change in its fitness via the intermediates of mRNA, proteins, and its traits. This concept, as we will see, forms the basis for how evolution works. To be clear, not every mutation is transcribed to mRNA. There are such things as silent mutations, and not every change in the mRNA affects the structure or function of the protein it codes for. And not every change in the protein changes an organism's traits. For example, maybe the protein isn't very important. And not every change in traits affects an organism's fitness. Maybe whether the finch has a pointy beak or a dull beak doesn't matter. Maybe it can get by with either. Even with these nuances, the point is that a change in DNA can cause a change in fitness. Not that it necessarily will. So here is the evolutionary tree of life. At the bottom is a universal common ancestor, abbreviated UCA. This was probably a single-celled organism, and it allegedly evolved into every type of living thing we know today, such as insects or primates. How does this evolution work? How does one use what was just described to change a species over time? Well, there are 3 steps to forming the branches of the tree. No matter how complex evolution is, it will always follow these three steps. Step one is genetic variation. So the genome of an organism must vary. The variation in the organisms must be genetic, or it won't get passed on to its offspring. For example, if a person has genes that make them strong, they might pass those genes on to their offspring. But if they acquire their strength by working out, then they will not pass that on. This rules out environmental influences as sources of variation. So variation must be genetic. This has two main sources. Source number one is through sexual reproduction. This is only for those species that reproduce sexually and does not include species that reproduce asexually, such as bacteria or archaea. This variation happens when sex cells, like a sperm or egg, divide through a process called meiosis. It is because of meiosis, particularly prophase 1 of meiosis, that each offspring is genetically different from its parents. This is done in a process called crossing over, in which fragments of DNA are randomly exchanged. This means that each sex cell is unique, genetically speaking. In this way, sexual reproduction produces genetic variation. This is why you don't look exactly like your parents, but a bacterium does. We reproduce sexually, bacteria do not. There are other sources of variation that feed into sexual reproduction. These include gene flow, which is the flow of genes between populations, having a small population, deliberately breeding certain individuals together, and being geographically isolated. All these feed into or enhance the effects that sexual reproduction has on genetic variation. However, this variation is restricted. There are limits on just how different the offspring can be because sexual reproduction only recombines genes that are already there. It does not form new genes. We can see this no matter how many times one breeds cows, as long as they are breeding with other cows, one won't end up with a fish or a goat. Cows stay cows, which is a problem for the tree of life because we all supposedly came from a universal common ancestor. This is why one needs another source of variation for evolution to work. This source is called mutation, or a change in one's DNA. Mutations are the raw material for evolution. There are many types of mutations, such as missense, frameshift, or insertion mutations, but what they all do is provide new genetic information, something that sexual reproduction cannot do. This allows for our common ancestors to potentially evolve into something completely different. So, mutations and sexual reproduction working together allow evolution to happen. And of course, for asexual organisms, it is just mutation. A key point is that both sources of genetic variation are completely random. The exchanging of DNA in prophase 1 of meiosis and mutations are both random events. Since this variation is the raw material of evolution, evolution posits that every living being we see is ultimately the result of random chance. Step 1 of forming the branches is to introduce genetic variation. Step 2 is natural selection. This is sometimes known as survival of the fittest, where those who are less fit die. However, an individual does not have to die for it to not pass on its genes. Nature only has to keep it from reproducing. So, natural selection is the process in which nature selects those genetic variants with higher fitness. For example, let's say there is a population of finches on an island, like Charles Darwin found on the Galapagos Islands, and because of mutation and sexual reproduction, they are just slightly genetically different from each other. This causes some finches to have smaller beaks and some to have bigger, stronger beaks. And let's say a drought happens so that the only remaining food is really big, tough seeds that one must break open with the beak to eat. This means that only the finches with the larger beaks can survive. The ones with the smaller beaks can't eat, so they starve. So the drought is a naturally selective event, and it exploits the genetic variation within a population. That is, if there was no variation, if all the finches had big beaks or if all of them had small beaks, they would either all survive or they would all die. Natural selection would have plenty of nature, but no selection. But as it is, the finches that remain all have large beaks. There are other methods besides natural selection that attempt to explain how populations evolve. The main other one is called punctuated equilibrium. We have two graphs here. The graph on the left represents natural selection, where the population gradually changes over time. The graph on the right represents punctuated equilibrium, where the population changes in sudden bursts. How could this happen? Well, the theory is that if a population gets really, really isolated, that they essentially inbreed to the point of creating a new species. And this all happens so fast that it leaves no fossil evidence behind. From what I know, punctuated equilibrium is largely speculative because it lacks fossil evidence, and because bigger jumps are less likely to happen by random chance. This is why it is not proposed as often as natural selection. Therefore, evolution by natural selection, the stronger theory, will be the focus of these videos. Okay, so we have natural selection acting on genetic variation to accomplish step 3 of our branches, which is a new population. That is, the population of finches before the drought had both large and small beaks. Now there are only finches with large beaks. Another example of this is the famous peppered moths, where more darkly colored moths were selected for because they were able to evade predators. The logic is that steps 1 through 3 are repeated over time, perhaps millions of years, to create a new species. So some of your large-beaked finches would have genetic variation, and another naturally selective event happens, selecting only certain large-beaked finches, and so on. You vary, select, change, vary, select, change, vary, select, change, thousands of times over millions of years. In this way, you would progress down the branch until there is a new species. So in summary, genetic variation is the raw material for evolution, natural selection is the force behind it, and a new population is the product. And to re-emphasize that key point, the genetic variation is completely random. So regardless of whether natural selection acts in a non-random way, at the bottom of evolution is complete randomness. There is no way around this. Okay, so the 3 steps explains the branches of the tree of life, but not the forks. I have explained how one species can change over time, but how does one get multiple species from the same common ancestor? The forks are produced in 2 steps. Step 1 is to split a population, perhaps geographically, like a mountain range. So maybe instead of starving, your small-beaked finches fly to a different island that has smaller seeds. Now each population can be exposed to a unique set of naturally selective events, and this means that 2 different species will be produced. A famous example of this is Darwin's finches. You can see the different species that were formed in the figure provided. This is more for your information, but this is an example of divergent evolution, which is a general term for a population diverging into 2 species. There is also convergent evolution, where structures evolve independently but do not share a common ancestor. An example is that both insects and birds have wings, but their common ancestor did not. So we have our 3 steps causing the changing of the populations. One then splits these populations geographically, allowing speciation to occur. So now that I have explained evolution in a nutshell, what is some of the biochemical evidence for it? Well, the main evidence is microevolution, or the adaptation that organisms make to the environment. Examples include the different breeds of dogs, such as a Chihuahua or a Golden Retriever. Or there are the peppered moths, where the darker colored moths were selected for. There are variants of the flu. The flu evolves each year, largely in response to antibiotics. This is why we have the yearly flu vaccine. And the logic is that we can see evolution on a micro level, so why not assume that over millions of years it could happen on a macro level? Then we have the genetic evidence, which includes junk DNA. This is DNA that seems to serve no purpose at all and is supposedly inherited from our ancestors. Then there are the similarities in DNA sequences. You may have heard that humans have 50% of their DNA in common with a banana, and this supposedly shows that humans and bananas have a common ancestor, probably very early in the tree of life. There are other evidences that exist that will not be addressed in these videos. Non-biochemical evidence includes the fossil record, in which we can see the progression of life from one form of life to another. An example is this line of skulls going from monkey to caveman to man. On the same line of reasoning, there are fossil intermediates, such as Archaeopteryx, which is the supposed missing link between dinosaurs and birds. Then there are the dating methods, which allow enough time for evolution to occur. I'm not talking about blind dates or double dates, but more like radiometric dating. For example, rubidium decomposing to yield strontium is used to date the Earth to about 4 billion years old. And there is the age of the universe, derived in part from the Big Bang Theory, which is thought to be about 13 billion years old. It only takes one chance for evolution to work, and there are a lot of chances in 13 billion years, the evolutionists would say. So even if evolution is unlikely, it is bound to produce something eventually. Then there are vestigial structures. They are the anatomical equivalent of junk DNA and include structures like the appendix or the tailbone that seem to serve no apparent purpose. Why else would we have them, the evolutionists would say, if they aren't inherited from our ancestors? Please note that this is not a comprehensive list of the evidence for evolution, but I think I hit the main points. Moving forward, I will only be addressing the biochemical evidence. Now that I have summarized Darwinian evolution, let me quote Charles Darwin, who said in The Origin of Species, "If it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous, successive, slight modifications, which we now know to be mutations, my theory would absolutely break down. But I can find no such case. In order to have these slight modifications, evolution must make three major assumptions. Firstly, it assumes that there are a virtually infinite number of naturally selective events. That is, genetic variation does nothing to evolve a population unless there is a corresponding event to select for it. And this number needs to be very, very high, or we would not have the diversity we have today. It also assumes that there is a nearly infinite number of organisms to work with. Why? Because the vast majority of the organisms in evolution die, or at least aren't able to reproduce. They are weeded out by natural selection. That is, every change in a population comes at the expense of those who do not have the change. So if there aren't an extremely large number of organisms, there won't be enough individuals left after a few generations of natural selection taking its toll. Generating these kinds of numbers isn't a problem for bacteria, which is why we see them evolving more readily. But it is a problem for larger organisms like blue whales, whose population is limited by the amount of resources the Earth has to offer. This, and because of the differences in complexity, is why we see larger organisms evolving less than things like bacteria. So, as one climbs the tree of life, evolution runs into a logistical problem. The third assumption, and the one that is to be the focus in these videos, is that a virtually infinite number of intermediate states exists. These are the stepping stones between different species, such as Archaeopteryx being the bridge between dinosaurs and birds. But since evolution occurs in such small steps, a virtually infinite number of these intermediates needs to exist. As we will see, biochemistry has much to say on this topic. We could add a fourth assumption, which is that a very large amount of time has existed for life to evolve. The chances of forming a cell or having a beneficial mutation in any given case is low. Therefore, evolution requires many chances. This means millions, if not billions, of years. This is one of the reasons why dating methods that date the Earth as being younger than billions of years old are thrown out. They do not allow for evolution to happen. Making these assumptions allows the evolutionist to gloss over the details of how evolution works. This is a problem because the first step in showing that any theory is at least plausible is to give a mechanism for how it works. A mechanism that glosses over details is no mechanism at all. So that's the end of part one. Hopefully, you have a basic understanding of how evolution works and the assumptions it makes. Before we get into the biochemistry, in part two, I will show why evolution should matter to Christians. See you there.